Hello. I'm going to do a brief introduction to talk about the enzyme lab that we're going to do, so just take notes accordingly. Okay, so in this lab, we're going to be using the enzyme catalase, also known as turnip peroxidase. I'm going to be getting it from a turnip, so they're going to call it turnip peroxidase in the lab, but it's a type of catalase which is breaking down hydrogen peroxide into water and oxygen. So this is just hydrogen peroxide if I write it out in words. Okay, and you're going to read the lab. It talks about how cells, all living things, have the ability to do this because peroxide is a toxic chemical that we need to break down. So we're just using this as our enzyme, and this is the reactant and the products we're going to be using. Now, if I want to measure the rate of a reaction, I can do it in a number of ways. It sort of depends on the reaction I'm running. If I have a way to measure how much reactant is in the system or the test tube, I can measure the disappearance of that somehow. Or I could measure the appearance of one of the products over time and see how fast one of these appears tells me how much this reaction is occurring. If it was an exothermic reaction, I could also measure the amount of heat being produced to tell how fast this is occurring. What we are going to do is we're going to use this technique because we have a way to measure the amount of oxygen. And since oxygen is a product, we can put our peroxide in the tube, our substrate, right? So remember, reactant and substrate are the same thing, really. We call it the substrate when it's reacting with an enzyme. So we're going to put the substrate, the hydrogen peroxide, in the test tube with the enzyme, and we're going to let the reaction go. But we're also going to put something in there called guaiacol. Guaiacol is an indicator. Indicator is just a chemical or something that will turn color in the presence of a certain molecule. So guaiacol that we're going to use is an indicator for oxygen, and it will turn brown in the presence of oxygen. So the darker brown our test tube turns, the more oxygen is produced, therefore the faster the rate of the reaction. So that's pretty simple how we're going to measure it. We're going to put guaiacol in the tube, we're going to put enzyme in the tube, we're going to put peroxide, our substrate, or reactant in the tube, and we're going to let it go and measure how brown it turns to see the rate of the reaction. So there's a couple ways to measure rate of reaction. You're going to read this in the lab. This is actually a cutout directly from, I think, the second to last page of the lab. But there's basically two ways we can measure rate. We're going to do it in kind of two ways. We're going to do both. Okay, so we're going to do both. We're going to do this way and this way. But one is what we call the relative rate of reaction. The relative rate of reaction. And that's going to be using a color palette. And I'm going to show you a picture of that here on the next page. So to measure the relative rate of the reaction, we're going to be using a color palette. You're going to have this picture. You're going to be given this exact picture, I believe, in lab, a little laminated copy of it. And this is a pre-done sort of color palette. This was pre-done to show you kind of the different colors of brown that it could turn, okay? And so you're going to basically you're going to have your test tube, here with your stuff in it, and it's going to be turning brown or like some color of brown. And then you're going to decide, okay, it's about this color. And we're going to number these just so we can give it a number instead of saying a little bit brown, sort of brown. Our color palette is going to be numbered, and so you will record that on the color palette, your relative rate of reaction was a 7, and that's relative to this color palette that was created by me prior to class, okay? So you're not going to have to create that color palette. I've already done that. It's just sort of a rule or a guideline we're going to use for how dark the brown is. The problem with this is this just tells us the relative rate of reaction. So based on when I made up this kind of color palette and took these pictures to give you, I assigned this color a 2 and this color a 3 and a 4 and so on. But if you're recording a 7 on this color palette, okay? I mean, it is quantitative in that I'm giving it a number, but it's only relative. So you can tell in your reaction, you might have one that's a 7 and another one that's a 10. And using this same color palette, you can tell this had a faster reaction rate than the 7, right? So like when we go, this is going to be a faster reaction rate. The darker it turns, the darker brown. So you can tell in your own experiment just relative to each other, but that doesn't mean anything. Those results are not universal, meaning if I report a 9 or a 7 or whatever, and you're comparing that with somebody else that's comparing this reaction in some sort of a research project or something, what's a 9 for us in our color palette that we made may not be the same 9 around the world, okay? So we can only compare a change, an increase, a decrease, or if it stays the same, sort of just in, and only sort of in our own kind of lab and the color palette that we created. But we're going to do this. So you're going to mix your stuff in your tube, and you're going to give a number using a color palette. And we're basically doing that as a backup. The other rate of reaction is down here. The other way, and the better way, is using a spectrophotometer, which, by the way, we also call it a colorimeter, spectrophotometer. It's the same thing. And it's an actual measuring device that's going to measure, we're going to measure the absorbance, okay? But we'll see it can also measure something called transmittance, and you might have used this in chemistry. And I think I talked about it over the summer. And it's going to give us the absolute rate of the reaction. So it's going to give us a number that's going to be the same for everybody. So let me skip down and talk about this. So the absolute rate of reaction. This is a quantitative way to measure how dark a color is. And the results are universal. So if I get an absorbance of 0.83, that's going to be the same. A 0.83 absorbance means the same everywhere, okay? Again, so we're going to, just in the lab, you might see it called a colorimeter, but it's the same thing. So when we measure what a spectrophotometer or a colorimeter does, you can see this isn't what ours look like, but it's basically a device here. And you have a little cuvette. We call this a cuvette in the lab. It's a little square tube that you're going to put your liquid in. And then you put that down in the hole here. And inside of here, there's a light source. Okay, so a light is going to be shined through that liquid. And based on how much light it gets through, there's a detector over here that will detect how much light transmits through. Okay, so I'm going to switch. I have a little blank here. I think I drew you guys this picture or something similar to it over the summer, but I'm going to real quickly just kind of do this again. So this is how a spectrophotometer or a colorimeter works. Okay, so it's a quantitative way to measure a color change. Okay, so if I had a test tube, again, you're going to have a test tube, or you're going to have a cuvette, and let's say that it has let's say your liquid turns really dark brown, okay, because let's say the reaction rate is happening a bunch, and you're going to have a light source here once it's in the spectrophotometer that you're going to shine through it. If it's a dark color, so if you have a darker color, whatever the color is, this is going to be brown, then what's going to happen is the light that's transmitted through will be kind of a lower number if you have a darker, okay? If the opposite of that, if I have, a lighter color brown, so let's just say it's like kind of light brown, I don't know how to do that, and we shine our light through it, you're going to have more light transmitted. You're going to have a higher, what we call transmittance, here. And remember, there's a detector sort of, kind of here, sort of a detector in the spectrophotometer that's detecting how much light gets through. And based on that, so the opposite of what we call you know, the light transmitted, which we call this transmittance. Okay, higher transmittance, this one will have a lower transmittance. You could measure the transmittance as a way to tell how dark it is. All right, so again, this is a lighter color. But the opposite of transmittance is something that we call absorbance. So let me do that a different color here. I don't know, let's just do purple. So, well, no, I'll just do this. So if less light is transmitted through, there's also some of the wavelengths of light are absorbed. So we call this the absorbance. So when you have a dark liquid like that, you're going to have less light that gets transmitted, and you're going to have more or a higher absorbance number. We're going to see that your absorbance and your transmittance, so your absorbance, what's being absorbed by the liquid, the wavelengths of light that are being absorbed, and your transmittance, those are going to be inversely related. Sort of, not directly, but so these are sort of inversely related. Okay. So we'd have the same thing down here. If I have a lighter liquid and when the light shines through, it's going to have, so there's going to be some light absorbed from the pigments in the liquid, but this is going to have a lower absorbance. So if we just look at, let's look at absorbance here. So this one's going to have a higher absorbance. This one's going to have a lower absorbance. This one's going to have a lower transmittance, and this one's going to have a higher transmittance. So when we measure in the spectrophotometer, we can either decide to take the absorbance number or the transmittance number. It's just a number that will basically tell us how dark the liquid is, okay? So that's how the spectrophotometer is going to work. And we're going to be dealing with brown. So you can see here absorbance, as the absorbance increases, the transmittance, here's absorbance, here's transmittance. They're kind of inversely related. It's not a direct line, but they're sort of the opposites of the other. So in our lab, we're going to be measuring the absorbance. I don't know why, we just chose it. So keep in mind, if you have absorbance here, the darker the brown it gets, this picture is blue, but the darker the brown, the higher the absorbance it's going to have. Okay, so we're going to be recording that number as well, okay, from our spectrophotometer. And this is going to be our absolute rate of reaction. So going back here, you're going to measure both, okay? You're going to measure the rate of the reaction using the color palette. You just look at the picture and pick what number you think it is. And then you're going to also put a sample of it into the cuvette and into the colorimeter and get an absorbance reading, which will be the absolute rate of reaction. And probably, again, assuming this works, sometimes these machines don't work. Assuming this works, this is what we're going to graph because it's better. It's a better way to measure, more universal, okay? Okay, so just a couple of things here. These are things this slide I'm actually going to have up when you're doing the lab. So some of this doesn't really matter, but glycol, the indicator that turns brown, is hazardous, so I'm going to have it in the fume hood, and we don't want to be breathing it. We're going to use pipettes. I'm going to kind of go over the procedure here. This doesn't matter. But one thing you have to do with the spectrophotometer is you have to do what we call it blanking it or calibrating it. And you're going to have directions for these on the lab day. And you're going to, kind of plug it in. There's going to be a blue sheet that has directions. You're going to have to set the wavelength right. Because depending on what color you're using, we're going to be using brown. You have to set the right wavelength for it to absorb or measure the absorbance. It's nanometers. But you must create a blank first. And so what I'm going to have you all do, you want to know just like the base liquid, what would be the absorbance of just the liquid that we're using? That's just enzyme and hydrogen peroxide. Because the enzyme we're going to see is a little bit of a cloudy mixture, because I'm just going to put a turnip in a blender and give you some juice. That's going to be your enzyme. And so if the base of the reaction, without it even reacting yet and turning, without glycol in there turning brown, we need to know sort of what our baseline is. And so that's what we are going to use to blank our spectrophotometer before we even start. So I was going to just kind of draw. You guys are going to read the directions. I kind of like to do what I call cookbooking it, and the directions are kind of confusing. So I'm just going to give you sort of an overview kind of lab day of what I want you to do. So the first thing you're going to do is kind of prepare, just kind of take these notes, prepare the colorimeter. Okay. Basically, we're going to blank it and turn it on. So when you come in, and this will be, I think this is on like the last page of the lab. You can read the directions in words. But basically, like I said, you're going to plug it in because it takes 5 minutes to warm up. And you're going to follow the directions on a blue sheet that I'm going to give you in lab. You're going to set the wavelength at 430 nanometers. And I don't really need to write through all this because you can follow the directions. I'm just going to kind of show you kind of in a picture what you're going to do. So once you plug it in and it warms up, and you're going to follow the directions on the blue sheet, which tells you how to set the wavelength through 430, you're going to create a blank. So you're going to take 2 milliliters of hydrogen peroxide, and you're going to take 1 milliliter of the enzyme solution that I give you, the turnip peroxidase. And you're going to just kind of mix this. You can just mix it in a little cup or a beaker. I'll have these for you. Okay. So just you're going to kind of put those two together and mix it. And then you're going to transfer it into a cuvette. Cuvette is just it's the little square thing that fits into the colorimeter. You can't put a beaker or a test tube directly in there. So cuvette, this is a cuvette. Okay, so you're going to transfer this liquid kind of into here. Except it won't really be a color because it'll be a little cloudy. And then you're going to put that in, you're going to have your spectrophotometer over here, or your colorimeter, and there's going to be a little hole kind of to put it in. You're going to put the cuvette into there. Here's our colorimeter. And then you're going to follow, there's a way to put it in there. You have to make sure the clear side's facing the arrow. And you're basically going to follow the directions, and you're going to hit a button. You're going to hit the calibrate button. You're going to hold that down. And then when the red light flashes, you know that it's ready and you're done. And then you're going to release the button. So this is just going to be our blank, basically. And you're going to have to do that before we even start. Just get our colorimeter. So that'll be, at that point, the absorbance, and we're going to, should read, there's going to be a reading on here. You'll be able to read, like, absorbance. There'll be like a little reading. The absorbance should be, around 0. We're pretty close to that. So it's just like blanking a scale when you put like a weigh boat on a scale and you hit the 0 button, because you don't want the weight to include that. It's sort of like the similar thing. So if this liquid is a teeny bit cloudy, just because of the enzyme, whatever, that's going to be now considered 0 and 0 absorbance. So when you come in, like one of you can do that in your group. The other thing is we're going to use pipettes, and you're going to label pipettes with a Sharpie, because I'm going to have all of these in a box for you. So they're going to be these 10 mil pipettes. These are going to be 10 mil pipettes. They kind of look like that. I'm terrible at drawing. But you're going to label one E. You're going to label the other one P, which I'll explain what these are. One NB. This is what the directions of the lab say. And one S. Okay, so E, this is just your enzyme. This is the pipette that you will use every time you're putting enzyme in something, okay, which is your ternary peroxidase. P, I hate this, it stands, they used P because P is supposed to stand for product, but what this really is the guaiacol. This is the guaiacol indicator that will test for the product oxygen. So that's just the way, if you want to label this, well, they call it P in the lab directions. Just keep in mind P, every time it tells you to use guaiacol, you're going to use this pipette. NB stands for neutral buffer. So I'm going to give you a whole bunch of different buffers because what we're going to be testing here is pH on the enzyme eventually. So you're just going to use the pH of 7 buffer. Whenever that's going to be used for neutral buffer, and then S stands for substrate. So you'll use that pipette every time you're putting hydrogen peroxide in a tube because we don't want to mix tubes. So it tells you just to label those, and if you remember, if you've ever used them, we're going to have those little, it's like a pipette pump that you put on the end, and you can switch the pump and the pipettes out as you're using them. Okay, so it's going to kind of tell you to do that. These directions are, if you look at page 8 in the lab, it kind of explains just all of that a little bit more. And then we're going to do 2 parts of the lab, and this is where it gets really confusing. So part 1 of the lab is everybody's going to do, when you guys are going to be in groups, and this is going to be, everybody's going to do a baseline reaction, which is basically measuring the reaction rate at a normal pH of 7. Okay, so I should have mentioned, if you haven't read the lab already, but going back, what we're going to be measuring here, here's kind of one of the pictures from the lab. We're going to be altering the pH. That's going to be like our independent variable. And we're going to measure the rate of the reaction. Let me go all the way back because I meant to write this at the beginning. So kind of what we're going to be measuring, our independent variable is going to be pH. That's what we're going to alter. And our dependent variable is going to be the rate of the reaction in response to pH. But we're going to measure that by, absorbance and the color palette. So on your graphs, you're actually going to be graphing the absorbance versus the pH eventually. But before we even do the real reaction, where we alter the different pHs, everybody's going to do a baseline reaction, okay, at the normal pH. It's sort of like our control group, right? So we're going to do a baseline reaction, measuring it at normal pH. Again, we can think of this. This is sort of like our control group. We want to see what the rate of the reaction is just normally. So I'd like to draw like a little picture. These directions are found on page, if you want to read pages 9 and 10 to kind of go along with this picture. I think the picture helps. So it's going to, if you read it, go ahead and read page 9 and 10 if you want and pause, or read it after this. It's going to be kind of confusing, but basically here's what you're going to do. You're going to have 2 test tubes. One, they tell you to label SPNB, and one, they tell you to label, ENB. Okay. And you're going to actually write that directly, again, with a Sharpie. I'm going to have you write right on the test tubes. And in this one, you're going to put 2 mils of hydrogen peroxide, your substrate, right? 1 mil of guaiacol. That's your indicator. And 1 mil milliliter of neutral buffer. That's why they call it the SPNB tube, because S is your substrate. This is your, right, this is just S. This is what they call P, but remember I said it's really glycol, the indicator for the product. And then this is your NB, which is your, this is going to be your pH 7 buffer. Okay, so you're going to put that in that tube. And then over here, E and NB, in this tube, you're going to put 1 ml of enzyme, which I will give you, that I will get from a turnip, and 3 ml of also neutral buffer, pH 7. So everything's at a pH of 7. Think about why we put this separate. As soon as, okay, we want these separate because here's my substrate, right? My peroxide. Here's my enzyme. As soon as I put them in the same tube, the reaction's going to start. So they have you starting out putting kind of them in 2 separate tubes, and then you're going to mix them. When you're ready, you're going to mix these 2. And you can just pour one. I think it tells you to use a pipette. You can just pour one into the other carefully. Okay? And as soon as you mix them, the reaction is going to start. Oops, I meant to. So you're going to mix those into 1 tube. Okay? And as soon as you mix, you're going to set your timer because the reaction's going to start. So you're going to have a timer. As soon as you mix them together, your reaction is going to start. This is going to kind of maybe start turning brown if the reaction is occurring, right? Because the glycol is going to be reacting with the oxygen. That's a product. As soon as you mix, you're going to immediately, because you got to get ready to take your results every minute, you're going to immediately transfer, that's going to be in a big test tube, approximately 3 ml, and you can just do 3 ml. So you're going to immediately take approximately 3 ml and put it in your little cuvette. And you're going to put the cuvette into the colorimeter that you've already blanked out. So you're going to put the cuvette into there, okay? So immediately you're going to do that, and you're going to then take readings every minute for 5 minutes. So, but you're going to be taking 2 readings. You're going to see the absorbance. There's, kind of a grid. You're going to see this absorbance number is going to start just going up as it turns brown. And you're going to be taking 2 readings. You're going to have some liquid left in here. You're going to have some liquid left in there because you're only going to transmit like 3 ml of it into there. Okay, so you're going to be reading the absorbance and looking at this and using the color palette. And every minute you're going to take so every minute for 5 minutes. You're going to take 2 readings. One with the color palette and one, one you're going to take the absorbance number. You're going to write that down from there. And 2, you're going to use the color palette that I gave you, that picture, and look at the color there, right? Once the cuvette is in there, you can't see the color. So that's why we'll look at what's left in here. So you're going to write the absorbance number and the color palette number on your data sheet and do that for 5 minutes. Everybody's going to do that. Okay. And then I'm going to have you graph it, and we'll get into the lab later, but you're going to measure time for 5 minutes, and you're going to measure the absorbance, and you're going to get sort of a rate, and you're going to calculate the slope of the line and get the basic rate of the reaction at a normal pH. So that's part 1. Everybody will do that the same to get a baseline reaction rate at a pH of 7. Part 2, and this is where the directions I think are really confusing. It gives you all these test tubes and all these different things. But basically, I want you to understand this is where we're going to alter the pH. All right, that's going to be my independent variable. Okay, we're actually doing the experiment here. And we're going to do pH of 3, 5, 6, 7. I don't know why they chose these. They're not evenly spaced, but there's 1, 2, 3, 4. There's 6 different pHs. Everybody can't do all of that. We just don't have time. So probably for the pH of 7, I'm just going to have you, use your number from up here. So I'm going to have you use your baseline because we already did a pH of 7. And then I will assign you, or your group, like 1 or maybe 2 of these other pHs. We'll see how much time we have. And then we're going to pool all that together to get the data, and then we will graph it. But basically, it's going to look the same. You're going to do it the same way we did up here. You're going to set up 2 test tubes. The only difference is going to be, instead of putting in neutral buffer, you're going to put in, well, you're going to put neutral buffer in this one. But with the enzyme, instead of putting a pH of 7, you're just going to put in, I'm going to give you a whole set of buffers. You're going to put in the pH of 3 or the pH of 5. Do the same thing, mix it, okay, and measure the rate of the reaction. Okay, so I don't know if we need to really draw that, but it's going to be the same. You're going to have a tube, and you're going to put, again, in this one, you're going to put your 2 mils of peroxide, which is your substrate. You're going to put 1 mil of guaiacol. Can't spell guaiacol. And you're going to put 1 mil. In this one, you're going to put 1 mil of neutral buffer, which is pH of 7. All right. Then over here, you're going to put 1 mil of your enzyme. And this is where it'll be different. You're going to put 3 mils of whatever pH buffer I assign you. Okay. So I might assign you the pH of 3. So you'll put the pH 3 buffer in there instead. And then the procedure is going to be the same. You're going to mix those together. Once you mix, the reaction starts. You're going to take 3 mils, put it in a cuvette, put that in the spectrophotometer, and so on, and take your readings and see how brown it turns. Okay. So you're going to do the procedure the same way in part 2. You're just going to use a different pH buffer. And I might have you just do 1 pH, or I might have you do 2, and then we'll pool them all together in a chart in your lab that you will get that looks like this. Okay. So you can see let's say you're assigned a pH of 3. At 1 minute, you're going to write the number from the color palette and the absorbance on the spec. You're going to do that 2 minutes, 3 minutes, 4 minutes, 5 minutes. And then we can pool together everybody's data here. And then we're going to graph it. And what do we think this graph is supposed to look like? If this time I graph pH versus absorbance, which, remember, the absorbance is telling me the reaction rate, it should look like that, right? Because that's what we just learned in class. Anyway, so that's kind of an introduction to the lab. Do your best, read through the directions, doing the pre-lab, etc. I hope that helped.